Everyone, this is Katie Goldsmith with MACAN. We'll wait just a minute here or two to let folks continue to sign on to the webinar, and then we'll get started. All right, I've got 1:01 on the clock, so why don't we go ahead and get started this afternoon. Hi everyone again, this is Katie Goldsmith. I am the project manager at the Mid-Atlantic Regional Council on the Ocean, or MARCO. I also serve as co-coordinator for MACAN. MACAN's other co-coordinator, Grace Saba, is assistant professor at Rutgers University, and she is also with Marcus. She is in attendance today, and together, if anyone has any MACAN related questions at the end of the webinar, her and I can respond. We are presenting this afternoon the Using Citizen Science and Monitoring Acidification webinar, the second in a MACAN webinar series. So real quick, I'll just remind everyone of some upcoming webinars within this series. On January 9th, we have a webinar with Marjorie Friedrich. She's going to be talking specifically about multiple stressors in the estuarine environment, what drives changes in the CO2 system. She's going to be talking about some of the modeling work that she's been doing. So if you're interested in that, keep your eyes open for the MACAN listserv information for registration. Also on February 6th, we'll be talking about the Research Priorities Work Group and the work that that group has been doing and getting some feedback from the rest of the MACAN listserv membership. And then on March 6th, you'll have a similar opportunity for the monitoring plan that the work group has been pulling together. We also want to remind everyone that if you have any other ideas, or if you want to present on some recent work or projects that you've been doing, we're always open to hearing about additional opportunities that people want for sharing their information. So please feel free to get in touch with Grace and I at info@midacan.org. So for today, we have some great presentations scheduled. The first presenter is Parker Gassett. He is a graduate student at the University of Maine working on an effort to fast track the forthcoming EPA guidelines for monitoring changes in pH and associated carbonate chemistry of coastal waters of the eastern US to citizen science monitoring groups. Parker Gassett is working on this effort alongside the NOAA OA office, the EPA, MIT Sea Grant and Maine Sea Grant, the Connecticut Department of Energy and Environmental Protection, and the University of Maine and Cooperative Extension Office. Our second presentation will be Emily Rivest. She'll be presenting on a project titled A Citizen Science Initiative in Global Change Ecology in the Chesapeake Bay. Emily is working with two high school 2 high schools at a total of 7 sites to evaluate methods for data collection by citizen scientists. This project also seeks to determine what aspects of water quality affect oyster growth the most, and if these drivers differ by location. Emily Rivest is an assistant professor at the Virginia Institute of Marine Science, or VIMS. Her areas of expertise are ecological physiology and global change biology. Dr. Rivest is passionate about exploring what makes coastal marine invertebrates tolerant or sensitive to changes in environmental conditions. Much of this work addresses the big picture challenges of climate change and how it will affect economically and ecologically important species like oysters. And then our third presenter, Mara Diaz, is the Water Quality Manager for the Surfrider Foundation. We'll also have a presentation by Charlie Plybon, who's the Oregon Policy Manager for Surfrider Foundation. Mara leads Surfrider Foundation's Clean Water Initiative, providing support and technical expertise to Surfrider volunteers who are monitoring the water quality at their favorite beach or trying to solve water pollution problems in their communities. Mara will be talking about the Surfrider Foundation's Blue Water Task Force Volunteer Water Testing Program. Through this program, citizen scientists are providing critical water quality information to protect public health at the beach, raise awareness of local pollution problems, and to bring together communities to implement solutions. Finally, Charlie Plybon plays a key leadership role in statewide ocean issues for Surfrider and supports strategic engagement of their Oregon chapter network and policy initiatives, grassroots campaigns, and community-based projects. Charlie will be presenting on Surfrider's engagement in citizen science partnership, placing pH monitoring units in Oregon's marine reserves to study, educate, and engage the public in ocean acidification. The project is spearheaded by Dr. Francis Chan of Oregon State University and the Partnership for Interdisciplinary Studies of Coastal Oceans, or PISCO. And this brings together multiple community groups along the Oregon coast with the state's leading researchers, state and federal agencies to monitor ocean acidification. So we have a variety of really great presentations today. Each will take about 15 minutes. We have about 10 minutes at the end of the webinar for any questions. I'll be collating those questions from the questions box on your control panel, so feel free to type those in at any time during any of the presentations, and all participants will be muted otherwise. A reminder that this webinar is being recorded and it will be available on the MACAN website on our resources page with the previous webinar series. So without further ado, we'll change it over to Parker and we'll get started.
All right. Well, thanks everyone for joining the call today. And also, I just really want to extend my gratitude for everyone who's involved in these coastal acidification networks. Keep up the good work. It's great to be here today, and your dedication and your leadership to this issue is so valuable. So I'm just involved with a small piece of this effort, but I'm happy to tell the story. And I just want to start with the big picture as an intro here. So a changing atmosphere is a problematic hallmark for humanity's influence on the globe, and it's just one of so many systemic changes that really demark our emergence into the Anthropocene. So we're in new territory in Earth's history. Initiated by human activity, we're seeing entirely novel changes in trophic energy flows, the nitrogen cycle, the carbon cycle, geologic processes. We are tinkering with the planet. And the first rule of tinkering, excuse me, is not to lose any of the parts. So this is the big picture of where we are right now. And ecosystem disturbance and mortality, these are well-known but not trivial challenges in the human-affected marine world. Eutrophication, anoxia, rates of disease, harmful algal blooms. These are areas where research is informing management in real time. And we're not there yet with ocean and coastal acidification. Now, ocean acidification was first mentioned only about 30 years ago, just for some context on that. And research on OA in that time has made enormous strides, but the field of coastal acidification and the nuances of the nearshore environment this field is really in its infancy. And I'll argue that the field at this stage in its life is also most able to learn new languages. And the language that I'm so happy today's webinar is discussing is the language of citizen science and a distributed monitoring system. So the dynamic environment of coastal acidification, the multiple drivers on estuarine and nearshore carbonate chemistry, the importance of saturation state, these are each the cutting edge topics being tackled by this field. And this talk's going to explore the opportunities to engage citizen science with this research. And for my presentation, it's grounded in the capacity of the Northeast citizen science groups to contribute to our scientific and our management understanding of OCA. Now, rapidly, there are improving and available instruments and methodologies for monitoring the marine carbon system. And our effort through this project aims to support a mission toward that distributed and excuse me, democratized community monitoring. So getting the tools and the support to the communities that are able to act on the behalf of a changing environment and able to observe a changing environment, that's really our set of goals. So the motivation for this work comes from the forthcoming release of EPA guidelines for measuring changes in seawater pH and the associated carbonate chemistry in the coastal environments of the eastern United States. Adam Pimenta and Jason Greer are the EPA scientists that are helping to galvanize and specifically to standardize efforts for how we collectively look at coastal acidification. And they're doing this by supporting fundamental improvements in how we measure and monitor these changes. So a lot of people are excited about this document. It's in review, so you'll have to stay tuned for its public release. But the guidelines explore a spectrum of methods. And most importantly, they highlight this weather versus climate data quality. And while some citizen science groups do have access to really high-end precision instruments, the range of short-term changes in the coastal carbonate system, that range warrants monitoring even from weather quality instrumentation. And this really opens the door for citizen science participation. And there's a general understanding of this hierarchical system of data quality. Not everyone needs to be looking at climate quality data to have meaningful research. And this type of distributed monitoring it'd be critical for vulnerability assessments, for early warning systems, and for establishing local relationships for municipal management to work on ocean and coastal acidification. So that's the context of why this might be important. And just a quick case for citizen science overall. The ecological questions that we now need to ask are rapidly expanding in complexity. And that's accompanied by a need for more information and larger data sets. And citizen science can provide a critical complementary dimension for research, and specifically a task force of involved people that might be unsurmountable by traditional forms of scientific inquiry. And also, within the research community for OCA, volunteer monitoring has the potential to address this mounting need for fine-scale research focused on particular localized acidification events or coastal hydrodynamics, and expert resources for monitoring and data collection can be scarce. So this really emphasizes the need for volunteer participation and low-cost data collection. And there's also this series of co-benefits related to citizen science as a methodology. Participation in this form of research has been reviewed to be a particularly impactful form of educational outreach. It positively influences participants' awareness and understanding of important scientific concepts, but also their attitudes and their behaviors regarding the scientific process overall. So formalizing citizen science programs to work in collaboration with research and with management needs, this has pragmatic importance for our monitoring capacity itself, the number of people involved with research, but also with this series of co-benefits regarding community participation with environmental resource concerns. But citizen science programs are not effortlessly available. It would be wonderful to wave a magic wand and have lots of folks meaningfully involved with research, but these projects are often challenged to find scientific methods that are useful for hypothesis testing while also remaining appropriate for an often volunteer experience. So that's the challenge of having these citizen science programs really be effective. And data management and research inconsistencies have been identified as the primary barrier for citizen science collected data to be used by formal researchers and to be used within the peer-reviewed academic literature. But we're not working from scratch here. There is an amazing community of stewardship organizations, the Friends of groups and the community water monitoring programs. And these programs and partnerships have long been researching various parameters of water quality. And these are the organizations that are integrated with their communities. They often have developed relationships with local and state agencies. They're committed first and foremost to understanding and advocating for the stewardship of their home environments. And it's these groups that are best positioned to realize citizen science programs for the marine carbon system and to tackle this specific aspect of the challenge of coastal acidification. So my work is specific to the Northeast, but these Friends of groups and community water monitoring programs are throughout our nation and really an untapped resource. So our first assessment in the Northeast found that over 50 groups are actively engaged with community or student or commercially oriented water monitoring efforts. So this is an untapped capacity. And our work began in the spring of 2017. It was kickstarted by a grant from the NOAA Ocean Acidification and Education Office. And the primary question was, what would it take to get the EPA guidelines information out to existing water monitoring programs in the Northeast? and what additional resources or support might benefit these groups to help their work become most impactful or for them to begin monitoring acidification for the first time? So we assembled a group, and as was mentioned before, with representatives from the NOAA office, the Environmental Protection Agency, MACAN, the Northeast Coastal Acidification Network, the Maine Sea Grant, the MIT Sea Grant, University of Maine, and our Cooperative Extension Office, the Connecticut Department of Energy and Environmental Protection, and also received support from the Senator George J. Mitchell Center for Sustainability Solutions. And this past summer, in our project planning phase, we identified a framework for workshops and webinars in Connecticut, Massachusetts, and Maine. And we're using a train-the-trainer model where small groups that we work with directly will be able to pass on information to a broader local constituency. So the goal is that in the spring of 2018 to facilitate connections and discussions through these webinars and workshops over coastal acidification monitoring efforts overall, plus to have the workshop days themselves aim to engage hands-on training with equipment and intercalibration, and have the training day itself transition into a working meeting about sharing data and research coordination among these groups. So just in the past couple of months, we've actually reached out to these citizen science organizations, and we did this by region and through surveys, and we're gratified to see that 100% of these organizations, they said they're interested in learning more about the coastal acidification system, sampling for the marine carbon system, and also coordinating research with others. So there's enthusiasm for the project overall. And early on in this work, we recognize that folks are going to be at different stages. They have different monitoring capacities, different instruments, different staff resources, different levels of interest overall. And we want to meet people where they are, give them some useful tools, hopefully inspire some to get involved with this issue for the first time. And we asked in our survey about individual missions and goals for their citizen science program, and dug a little deeper into what these groups are already monitoring. And that really acted as a springboard for us to get a better sense of what it is we need to provide through these series of trainings. So I'm just going to highlight a little bit of our preliminary results from this survey series, just to give you a sense of the direction of our project right now. So, for example, we asked people, which resources would you like to have? And at the very top of the list was, a workbook, a toolkit describing the recommended protocols for research. And this would really be an extension of the EPA guidelines. So the EPA text, though it's forthcoming, is broad and relates to a number of different aspects of research that are not all pertinent to a citizen science level organization. And so this type of citizen science specific workbook would kind of be a synthesis of the most important aspects of the EPA text and a little bit more user friendly to get folks involved. And of course the EPA text would still exist for further reference, but this was kind of a key resource that was identified. And next was this need for local assistance and local science advisors. So technical assistance, calibrating equipment, cross comparisons with laboratory samples. Just beneath this is connections with local science advisors to field questions and help interpret research results. So we saw this need to develop connections between either university staff or professional research laboratories. And we reached out to a technical advisory team and are starting to get them involved with this workshop and webinar series and hopefully foster the early stages of connections between these groups. And in some places that collaboration already exists, but specific to coastal acidification, our goal is really to help kindle those relationships. And the next set of resources were all education and outreach oriented. So pamphlets or sample editorials, ways to communicate about coastal acidification in a way that really makes this issue relevant to localities. And so this is kind of just a sampling of the types of resources that we're aiming to provide as a component of the workshop series in the future. So I'm just sharing this to give you a little bit of a sense of, what is the scope and scale of our vision. This project's in its early stages, but this is the direction that we're taking it. And kind of just to summarize, it's the very first time that we're anticipating EPA guidelines for coastal acidification. And that's an important step in galvanizing this research community overall. And we see this tiered monitoring option where weather and climate quality data are both important for our current stage of research. And that really opens the door to citizen scientists being involved with this research topic. And also, citizen science has this co-benefit not only of being able to sample and survey such a large-scale area that's totally heterogeneous for different coastal acidification conditions based on estuary or nearshore environment dynamics, and also that citizen science involvement with this issue has these co-benefits of more people involved with environmental resource concerns, a broader base able to establish the connections between research and management. So we're moving forward with workshops in the spring, and I just want to thank MACAN again. It's so exciting to have this dialogue between the coastal acidification networks, and lots of opportunities for MACAN to get involved, resources that you have that we might want to share with our constituencies, or for you all to be able to jump on board this type of project in your area. So with that, I'm going to pass the torch and just say thanks again. We're really excited about this work and happy to share about it with you all.
Thanks so much, Parker. That was great. That I think we'll definitely be interested in hearing as the project progresses and hearing about more of the results. But thanks so much for kicking us off here. We'll shift it over now to Emily or Dr. Rivest. And again, if folks have any questions for Parker, please go ahead and type them in the questions or chat functions on your control panel, and we'll be answering them at the end of the webinar. Take it away, Emily.
Thanks so much. It's really a pleasure to be here today to talk with you about a project that I started shortly after I arrived at VIMS. And this is a little bit different perhaps than the perspective of the other panelists today. And I'm a faculty member really interested in exploring the possibility of using citizen scientists in my research. And so this is this project that I'll present today is kind of a first stab at that, specifically related to ocean acidification and carbonate chemistry in the Chesapeake Bay. So I first have to acknowledge the many people who have made this project possible. As Katie mentioned in the introduction, I'm working with two high school teachers, Sarah and Liz, who are instrumental for making sure the data collection happens, and that's facilitated by some education and outreach coordinators. There are many students at VIMS and our affiliated institution, William and Mary, who are involved in analyzing samples and pictures. And of course, the funding comes from the Dominion Energy Foundation. They have a specific grant for environmental stewardship here. And the two schools that we're working with, I should mention, are Chesapeake Bay Governor School and Cape Henry Collegiate. So in my research program, I'm really interested in biological consequences of anthropogenic change in coastal ecosystems. And so one interaction involved in the way I think about my research is this interaction between long-term environmental change and natural environmental variability. So when we think about this interaction, we want to know what will habitat conditions look like in the future as this sort of long-term change happens on top of these very dynamic coastal regimes that we have already. And we know that coastal marine environments have natural variability across space and also across time. And so I'm interested in that and how that affects the biological response of animals like oysters to environmental conditions. Does that natural variability matter? And then how can that inform the way we think about how these animals will respond to future environmental change? But in the sort of evolution of the field of global change biology, a lot of what we know about how these animals will respond to future environmental change, like ocean acidification, has come from laboratory experiments where, to grossly simplify things, we place animals in a bucket and change the water chemistry and see what happens. But as we've learned more about this natural environmental variability, we need to be able to understand, are these processes already occurring in the field, and what the interaction might be. So there's this need to really link the lab and the field. And that's where citizen scientists could come in. So given the high degree of spatial variability in carbonate chemistry within the Chesapeake Bay, for example, we really need this sort of merging of efforts to assess water quality and also efforts to assess oysters, their abundance, their diversity, their performance. And when I arrived at VIMS, I'm still learning a lot about the coastal science going on here. So if anyone has additional insights to share, I would love to hear them. But some of the major players that are, from what I've learned so far, that are measuring water quality and specifically pH are the National Estuarine Research Reserve that we have here, NOAA's CVIB buoys. There are some cruises that happen in the bay, and the Chesapeake Bay program does a lot to monitor water quality, but they don't currently measure pH, I believe, or carbonate chemistry. And then, of course, there are also efforts to monitor oysters. VIMS has a monitoring program run by Roger Mann, and the Chesapeake Bay program also monitors oysters. But frustratingly for me, I'm wanting to understand this link between the environment and water quality, that these data aren't being collected in a concerted way so that we have measurements of water quality and oyster performance happening at the same place at the same time, or other measures of biological performance for other species, that kind of thing. So that's what inspired this program, is that I wanted to capitalize on citizens as a way to get more hands and feet in the water to help us really capture the difference in response of oysters across these spatial gradients of environmental variability. And so the program, this project that got funded, represents a citizen science approach to exploring the relationship between the environment and organism. specifically oysters. And so we're working with Chesapeake Bay Governor's School and Cape Henry Collegiate, as I mentioned, and there is a short video that you can find at the link here. We weren't able to show it through the webinar, but I encourage you to go look at that. It was produced by Dominion Energy to highlight the beginning of our project. So we have named the CSI Oyster, and it's a one-year project that was funded. And one of the goals is to evaluate these different techniques for measuring water quality, including carbonate chemistry and oyster growth. And I'm excited to read the EPA guidelines document when it comes out and help and have that inform our project design moving forward if we can do so. But in the meantime, we're comparing these techniques on our own to see what works best for our students in terms of data quality, in terms of the cost of the analysis or the equipment, in terms of the ability to teach citizens how to perform the measurements. And then using these data, we plan to explore the relationship between water quality and oyster performance. So this is a map showing our 10 sites. We have 3 sites to the north that are being used by students at the Chesapeake Bay Governor's School. So those are along the Rappahannock and Piankatank rivers along the Chesapeake Bay. We have 1 site at VIMS where my volunteers, undergraduate students from William and Mary, collect data every week. And then we have 3 sites down in the Virginia Beach area close to Cape Henry Collegiate. And here are photos of the 2 teachers who are involved in the project, Sarah Beam, who's with Chesapeake Bay Governor's School, and Liz Moore on the bottom at Cape Henry Collegiate. And so the teachers involve their students in this project in a variety of different ways. So we hope to reach about 100 high school students throughout the course of this year. My graduate student, Annie Schatz, is involved in project management and analyzing water samples. I provide overall project management, and then our 3 education outreach coordinators help liaise between us and the high schools. And then, of course, when the high schools aren't in session, we still have to collect the data, and so there are other volunteers who fill in during that time. So one of the things that we want to do with this project is to compare water quality measurements. So at each site, we have a crate of oysters deployed, as you can see in the center photo here. And on the crate is a little hobo logger that autonomously records conductivity and temperature every 30 minutes. And then when the students are there, they use a handheld YSI to measure temperature, conductivity, dissolved oxygen, and pH. So you can see on the left. And then we've also trained them how to collect and preserve bottle samples, which then are returned to me at VIMS, and I measure pH and total alkalinity according to best practices. So in the end, we'll be able to compare all of these measurements and understand in terms of data analysis and understanding of the regimes of water quality, the trade-offs between the cost of the measurement, the frequency of the sampling, and other logistical constraints. And so, as I mentioned, we have the oysters in the crates. So when the students are there, they are also measuring oyster size, and they're doing that in 2 different ways. They use calipers to measure shell height, and they also take images of the oysters using the photo bucket method, which is something that I made, although it's not a unique concept. And all those photos get uploaded to a shared box folder, and everyone helps to analyze the images. And then for some of the sites, the schools each have a core site, which they visit weekly. And there are 2 other sites they visit monthly. So again, we can evaluate the trade-off of sampling frequency. So I'll just show you some of the data we have so far. We're almost 6 months into this project, so we don't have enough data to do some robust statistical analysis yet. But here is showing you a comparison of measurements of shell height of the oysters. So for each oyster, we're measuring shell height in 2 ways, and so we can compare how consistent those measurements are to each other. And from the data we've collected so far, we can say that if you measure an oyster with a caliper, it's within 0.3 millimeters of the size you would measure from taking its picture and analyzing it using ImageJ. So I'm very happy with that. And as we continue to go and the oysters grow, and this relationship might change, but we'll just have to wait and see. Here's just an example of some of the pictures that we take with the photo bucket, and this is of the, not necessarily the same oyster, but showing you a time series, I guess, of photos showing oyster growth over 4 months, which is exciting for the students to see as they go back every week or every month. And here is just a rough plot of how the oysters are growing so far. And you can see in the gray and yellow and blue lines that the oysters at our northern sites are growing the fastest. The one, Long Creek, the oysters died a couple months into the project, so that site is no longer continuing. But you can see some of the other sites like Bay Cliff, the oysters have very different growth trajectories. So we're excited to see how these growth trajectories might line up with some of the environmental variables that we're measuring. So I mentioned that the oysters seem to be growing the fastest at these more northern sites, and this is just extremely rough cut average of the YSI data that we've collected so far. And you can see that in general at these northern sites, the temperature is higher, the average pH is higher, the DO is higher, and the salinity is lower than at some of our other sites. So again, at the end of the project, when we do all this analysis, it'll be interesting to see if these patterns are the same or not. But that's what we have so far. So in terms of expanding our footprint of understanding about effects of OA using citizen scientists through CSI Oyster, we're focused on observing how oysters respond to seasonal changes in their habitats. And that will hopefully give us a better understanding of Virginia oyster habitats. We hope to identify drivers of oyster growth at each site, recognizing that they might be different, and sites that have the best water quality for oyster growth. This current grant ends in April. We plan to use the information that we gather with these high school students and the results to refine our procedures for future use. We're seeking additional funding. One of the ways that these types of data could be directly integrated into academic research is through model validation. So Mark Brush, who's a professor here, and I have been talking about using these data to tune his ecosystem model, which would allow us to predict ecosystem services generated by oyster restoration at these sites. So that's a really exciting kind of add-on of primary research that could come from this. And eventually we hope to train new teachers and partner with these, as Parker called it, friends of, but we have some citizen science organizations here already that are invested in oysters. So there's the possibility to partner with them in the future once we have our methods nailed down. So with that, I'd like to thank you again for listening, and I look forward to your questions at the end of the webinar.
Thanks so much, Emily. What a cool project. We're gonna shift gears now over to Mara and Charlie. And as Emily said, just a quick reminder to go ahead and ask questions into the chat or question boxes on your control panel. Thanks.
Great, thank you. So before I dive into our citizen science program, I thought I'd just give you a quick introduction into who Surfrider Foundation is. We're a grassroots environmental nonprofit organization. We're dedicated to the protection and enjoyment of our ocean, waves, and beaches. This is a map of where we have chapters located. Currently, we have a network of nearly 150 now, volunteer-run chapters and clubs that are set up in local schools. And basically who we are is we're people who love the beach. We're surfers, swimmers, beach goers, beach volleyball, people who just love the beach and want to do what they can to help protect it, make sure it stays there, it's accessible by the public, and the water's clean and safe. So one of the programs we run to ensure that we have clean water at the beach is our Blue Water Task Force program. This is Surfrider's volunteer water testing program. We have about a 20-plus year history running this program. We currently have a national network of approximately 35 volunteer-run labs that are providing water quality information aimed at protecting public health at the beach, raising awareness of local pollution problems, and bringing together communities so that we can solve those pollution problems. This is a map of where we currently have Surfrider volunteer labs testing right now. Here on the East Coast in the Mid-Atlantic region, we have a lab running out of Virginia Beach. We have one in New York City, down in the Rockaways, and one at the end of Eastern Long Island. At the core of this program, we have lab programs that are testing ocean and bay beaches and freshwater sources, such as creek and river mouths and stormwater discharge sites. We're testing for Enterococcus bacteria, as this is what the health standards set on to determine if water is safe for swimming or not. And we set up our sampling programs to fill in the data gaps that are left from the state and local health agency beach monitoring programs. So we're testing year round when those agency programs generally only cover the summer months. We test popular surf spots and sources of pollution. We have all of our data posted online as well as maps of our sampling sites. And because these are completely volunteer run programs, there's a lot of diversity in how each program is set up. Some of the chapters, run the entire program, the sampling, the lab work, all on their own, but more often than not, the chapters have other local partners. They might have lab space borrowed at an aquarium or a local university. Some chapters provide volunteers to other established watershed groups. And many of our chapters partner with local high schools. They purchase the equipment needed and the supplies. They go out and they collect the samples, and then the lab space is provided, in a classroom at the school. There's a teacher advisor advising students who then process the samples and read the results. This is an example of one of our lab pages on surfrider.org. Shows you the sites that are being tested and whether the bacteria levels on the latest testing date were safe for swimming or not. One of the great things about our site is it's very easy to share that information with social media, which many of our chapters have a pretty big social presence in their community, so it's a great way to get the word out on water quality at the beach. And beyond just providing that basic information on bacterial levels in recreational waters, this has been an extremely good program, successful for building community awareness of pollution problems, educating students and grooming future environmental leaders, and developing partnerships that chapters can work with to then find the sources of pollution and implement solutions, ultimately leading to clean water at the beach, which is what we're really trying to do. There's a lot of really good examples on Surfrider's website of case studies of ways that chapters have used their citizen science program to bring together communities to solve water pollution problems. If any of you want to dig a little further, you can follow this link or send me an email. But another program that I wanted to talk about is our SmartFin program. which is just been launched this year in May. SmartFin is a surfboard fin, which is used to measure ocean properties. Currently, we have sensors that measure temperature, location, and wave motion, but we also have additional sensors under development to measure pH, dissolved oxygen, and chlorophyll. And the way this works is a surfer has one of these smart fins in his surfboard, and when he gets to the beach, he taps on the fin 3 times to turn his sensor on, goes for a surf. It will stay on and make measurements for up to 2.5 hours, and then when the surfer gets out of the water, they tap the fin again to turn it off, and then they upload their data through a cell phone app onto the iCloud. And then our partners over at Scripps Institute of Oceanography access that data. And we're currently, it's not publicly available yet. We're currently working on coming up with the backend database and the website, which will allow this data to be all accessed by the public. And I guess I should have also mentioned, in addition to Scripps partnering with us on this, we are also working with the Lost Bird Project and Future Fins. We just launched it in May. We're piloting the project in San Diego, and what we really want to work through by working just with this one chapter first is identifying the tech issues and user barriers with the technology, trying to figure out a good distribution strategy to get it to surfers who are really going to use it and, you know, upload their data in a way that's most useful for our research purposes, and to help develop tools that will assist the chapter volunteers and the people using this technology to help build awareness of climate change issues in their community. So far, we have 50 of these fins out in use. There's a lot more surfers on a wait list waiting to use it. And like I said, with the super surfers and drifters, we're trying to really figure out how to identify those surfers who are really going to stick to the program and use the fin in the most useful way for us. And that is it from me. I believe now we hand it over to Charlie.
Yeah, can folks see okay?
We can, yeah. Thanks, Charlie.
Perfect. All right. I just want to make sure I'm like in presentation mode because I'm seeing 2 different modes here on my screen, but if everybody sees okay, then I'll go ahead and get started. My name is Charlie Plybon. I'm the Oregon Policy Manager for Surfrider Foundation. And we have a pretty big Blue Water Task Force program here in Oregon, which because of our Blue Water Task Force program, we have 7 different labs up and down the coast. And because of our relationships with scientists and local managers, we were attracted to a partnership with Oregon State University to better look at ocean acidification and hypoxia in Oregon's marine reserves. And so you're going to hear OAH several times because both in policy and in the management context, we look at acidification and hypoxia along the Oregon coast as mutually exclusive. But for the purpose of the presentation, I'll try and focus mainly on the OA measurements that we do. So just sort of a general outline of the presentation here, give you a little bit of management and policy context, given we're on the West Coast. And I think most folks on this call are probably on the East Coast, and what it's kind of about where we are in that context, why we monitor in marine reserves, how we selected our sites, and why we're actually monitoring there. So we'll talk a little bit about the monitoring itself, and then the value of partnerships and what comes with that, like outreach and engagement, some examples of that. So just kind of to start off, you know, over the years, we've been taking a lot of measures and looking at Oregon's nearshore in a lot of different ways. And one of the things that really jumped out at us from 2000 to 2000 to date is that, you know, we live in a very vulnerable and changing coastal ocean environment. Really, one of the big keystones for this change in ocean chemistry that we were noticing in Oregon was the hypoxic events that we have here, and those are low oxygen events. You can see that they really started to jump up in 2000 to 2005, and then in 2006 is when we had our really first big hypoxic event. And you can see on the right-hand side, these are just some snapshots from some ROV surveys. The photos up at the top represent the same areas that were later done during the hypoxic event at the bottom, and you can see the difference in sea life. So this was, you know, had some management concerns for us here in Oregon. And then when it came to ocean acidification, oysters were really our big warning sign. We knew, you know, Parker spoke of, you know, the history behind us looking at and thinking about pH and ocean acidification. But it was really Whiskey Creek Hatchery in 2007 who experienced changes in the seawater that they were pumping in from Netar's Bay that didn't allow them to actually raise their spat. So the larvae that they were raising in the oyster farm was actually unsuccessful. And it took them like 3 or 4 months to actually figure out that it was indeed an altered chemistry in the bay and lower pH that was preventing them from growing oysters. So this really moved ocean acidification to the forefront of interest on the West Coast, particularly in Oregon, as we were ground zero for some of these low pH waters. So it left us in a position of, you know, is this something that we wait and watch happen to us, OAH being ocean acidification and hypoxia, or is this something that we should get out in front of and try and do something about it? What can we do? Can we change the chemistry of the ocean? Probably not, but there are things that we can do to alter our management and better understand the impacts moving forward. So in 2013, to give you some policy context, the West Coast Ocean Acidification and Hypoxia Science Panel was convened. This is a group of about 20 different scientists from California, Washington, Oregon, Columbia, and the charge of that group was really to enhance our understanding of ocean acidification and hypoxia and develop a suite of options for decision makers to think about moving forward, which is an interesting place to put both managers and scientists into this sort of recommendations around policy as much around science. What came out of that was sort of this roadmap of local management options to address ocean acidification and hypoxia, primarily looking in 2 buckets, reducing our exposure and then enhancing the ability of biota to cope. So you can see there's several different sort of buckets of sort of management strategies and recommendations that would come out of this report. Zoning in on a couple of those, recommendation 5 and recommendation 7. One was, you know, advancing the adaptive capacity of marine species and ecosystems. So looking at the areas like marine protected areas where we have invested in some protection and seeing, you know, is ocean acidification and hypoxia, is that the Achilles heel of these areas? Or are these areas that have greater adaptive capacity to these sorts of ocean chemistry changes? The other piece to this was really a recommendation that went into, you know, building out and sustaining a West Coast monitoring program that sort of meets these management needs so that we identify low pH waters before they impact things like our marine ecosystems, our crab fisheries, our oyster farms. So a lot of that, you know, action was around defining the gaps between monitoring efforts and management needs. So at face value, you know, you're looking at the ocean, you know, this is the Global Ocean Acidification Observing Network. And when you look at this, you're like, gosh, man, we take a lot of measurements in the ocean. I mean, don't we have enough monitoring going on already? And, you know, when you look at that at its face value, it does look like we have a lot going on in our ocean. But if you hone in to, you know, just what's off of the Oregon coast, what is still taking measurements, what is actually looking at, you know, the full carbon suite and arriving at things like aragonite saturation, which give us a better understanding of, you know, ocean acidification. And then what's left in our state waters is we really had very few monitoring going on, not a whole lot going on. And so there were some big needs for filling monitoring gaps. And there's some barriers for that as well. So why marine reserves? Well, for several reasons, Oregon marine reserves were attractive to researchers for monitoring. One is that these are refuges where other stressors are sort of minimized. So we know from the academic research and otherwise that non-point source pollution, other things, runoff can sort of add to and increase stressors in the marine environment that may exacerbate hypoxia or ocean acidification. So these marine reserves and protected areas are areas that, where we minimize that. These are areas that are not developed and don't have those sorts of stressors. These areas are also intended to be somewhat of a reservoir of demographic and genetic diversity. So these were the resilient areas of our ocean and our near shore that we were investing in for protection. So it would be worthy of us to understand whether they are truly resilient to ocean climate changes that are happening. And then third, they just serve as really nice listening posts. They are great comparison areas for all the other reasons that I mentioned. And there's also generally a group of individuals, whether that's a Friends Up group or whether that's partner groups like Surfrider Foundation, the Nature Conservancy, that are invested in these areas. So there's NGO partners and community partners on the ground. So in 2015, late 2015, Oregon State University approached Surfrider Foundation about using the marine reserves as sort of these sentinel listening sites to better understand ocean acidification. And like I mentioned before, this largely came about because of our network of water quality monitoring that we do for bacteria that Mara spoke a little bit about. And they knew that we were well connected also to some of these community groups that do work in the marine reserves, whether that's citizen science efforts or whether that's just stewardship actions. So groups like you see on the far left here, the Redfish Rocks community team. This is like a Friends Of group for the Redfish Rocks Marine Reserve, and they are one of the partners in this project. So how does this work? What are we exactly doing, and where and how do we do the monitoring? The monitoring takes place in intertidal areas, and we have basically, or Dr. Chan has basically created a housing that we are able to bolt down to the rocks in the intertidal area and slide a pH sensor into it. This is a Durafet pH sensor, so it's highly sensitive. We're measuring real-time measurements for pH and temperature, and then using proxies for dissolved oxygen and other carbonate chemistry to get at things like aragonite saturation. All that stuff sort of happens in the lab, and that happens with folks like our academic partners in Oregon State University, like Dr. Chan or his lab rats. For us, the citizen science end of it is simply installing those units. Once those units are installed in the summertime, we go and service them monthly. So that means that we take the sensor out, we mail it back to Oregon State University, and we replace it with another sensor. So we just sort of do this rotation on a monthly basis. Those sensors go back to the lab, they get analyzed, the data gets downloaded, and then it gets interpreted back to us. And by interpreted, I should say they send us some graphs of what data is there. So how does this help the researchers? Well, for one, you can see from Cape Falcon up at the northernmost site down to Redfish Rocks, that's about 300 miles apart. So for a researcher on a monthly basis to go and service each one of those would take a considerable amount of time and gas and energy. So place-basing people that live in those communities that can just mail these back and forth is a high benefit for the researcher. It allows them to, more or less just mail things rather than be everywhere at once. For us, it allows us to engage people in the measurements that scientists are taking and using to talk about things like climate change. And this is really important to us as an organization. We believe that engaging people in the science, builds confidence in that science and builds confidence in the policies and the management actions that may come out of that. So it's a sort of a win-win relationship. And then I would also say that, marine reserves, because they have these community groups and other state agencies that are invested in these areas, they're hotspots for engagement. So these are just a couple of screenshots from stories that have been written about this work. And this is sort of, you know, elevating the work that the scientist is doing and the outreach that the scientist is able to do. So these are, you know, one's from the Oregon Marine Reserves program website. It's a story that was up there from the Oregon Department of Fish and Wildlife, another from our page. We have lots of blogs, news stories, videos. We're developing a school curriculum. And we're also sort of venturing into better storytelling. And this is a part of the project that I'm pretty proud of and hopeful that we can improve. It's sort of in its beta form, but right now that data that we get back from the scientist is essentially in graphical form, and we're trying to make that more meaningful to people. People don't necessarily relate very well to changes in the pH. And so we try and put those graphs and we put those sort of numbers as indicators around how they affect animals. And we storytell about animals on this storyboard website that we created. And I've got the web link up at the very top of this slide. You can see if you want to read a little bit more about that or find out more about what we're doing with that site. Again, it is sort of the place where we're trying to tell the story of ocean acidification in Oregon. Build on the citizen science initiative by interpreting the results, but also allow these communities to tell their story, why they're engaging in this science and why they care is as important as the results itself to share. And then lastly, I just want to say that there's a lot of folks involved in this work. Surfrider is really proud to be a leader in working with Oregon State University on some of this monitoring along the coast here in Oregon. But we wouldn't be doing this without a lot of different partners that have informed and been a part of this process and continue to move this process forward. And like I mentioned, there's this storyboard map and our ability to talk about the monitoring we're doing has really attracted more funding, more partners, and a lot of educational opportunities as well. So I will finish with that and leave it there.
Thanks so much, Mara and Charlie. I think folks, attendees today can understand why we had such a hard time picking just one or two presenters today. All four of these projects, all four of these speakers today and the projects they've talked about are so neat. I know I'm looking at the clock and we're quite short on time, so I'll just keep it to maybe two questions today. So the first question is, looks like it's directed towards Emily, Dr. Rivest. It says, Emily, how are your water samples for this project analyzed?
So the bottle samples that our students preserve, we've trained them and provide the necessary equipment to have them add saturated mercuric chloride to the bottle samples, just like what I do when I preserve samples in my research. And then they get brought back to VIMS, and we use spectrophotometric pH methods and titration for total alkalinity to measure the carbonate chemistry of the water.
Okay, thanks. Parker, are there any plans for the research group to develop any of the recommended resources for citizen scientists? I.e. the pamphlet, for example, that you had talked about during your presentation? Parker, you might be on mute.
Yeah, so great question, and that's definitely on our radar for this spring. And kind of the model that we're using is that we'll try to take a first crack at a lot of those resources, but ideally we'll endorse this co-creative process. So getting the actual citizen science groups involved with some element of contributing to these final products is really a goal for us, just to inspire that ownership over some of these resources. But certainly over the next probably 3 or 4 months, we're going to try to make a pilot version of at least several or more of these products. So great question, and feel free to reach out to me through email after this webinar also. Thanks.
Thanks, Parker. And I guess I'll just finish it off with Mara, how can people get on the wait list for a smart fin? And then also related to that, oh, there's a recommendation to spread this information through the cans once it's publicly available through the coastal acidification networks.
Yes, thank you. I think if you want to get on a waiting list for a smart fin, the best idea would be to email me for now. One of my colleagues manages this program, and I'll connect you with her. So that's mdias@surfrider.org. And are there plans for getting the data? First, we have to get the data up where it's publicly available first, I think, and then we can move towards, if we're talking about the SmartFin data, then we can talk about sharing it with other organizations like the CANs. I think, yes. I just think we're not quite there yet.
Okay, thanks. So I'm sorry we're not going to get to all the questions today. We had some great presentations. If you want us to get you in touch with any of the presenters that you heard today, go ahead and email us at info@midacan.org and we'll create the connection. Of course, feel free to email them directly, but if you missed their emails, we can definitely get you in touch. Thanks again so much for joining today, everyone. Thank you. Huge thank you to all our great presentations. And hopefully we'll hear from everyone next month on our webinar in January. Thanks so much. Take care, all.